Ontogeny recapitulates phylogeny. This is an incredibly famous saying among developmental biologists. It was coined by Ernst Haeckel, and you'll hear it over and over again if you take a developmental biology class, and we would be remiss if we didn't say it at least once whenever we're talking about a developing human. Now, we need to expand and back up from the developing human and talk about more species here, because let's break down that phrase. Ontogeny refers to this development of an organism, so the stages that we're going through and walking through now in this lesson. Phylogeny is much more broad. It's thinking about the evolutionary development of a species. And sometimes we talk about a phylogenetic tree in biology, kind of thinking about how all of us as living things kind of came from, one ancestor, and we divided and got more and more different. Plants went one way, bacteria went another way, animals went yet another way, and then the animals become even more varied and diverse. However, let's just take animal development. If we look at these embryos here, okay, and if you're not careful about how you interpret this figure, you might have already been a little bit fooled. Okay, this certainly looks like a bunch of embryos here. Okay. And let's just take a look at our human real quick from the beginning. This is about week 4, and we can see structures that we recognize. Right here is the head. We can see an eye developing there. I can see what looks like a spinal cord and some muscles coming along those primitive somites. I can see an arm bud and a leg bud, but then I also see things that are maybe strange to me because humans don't have tails. Pharyngeal arches, do you mean gills? Okay, so what's going on here? Let's look at this picture. You actually interpret this from top to bottom. Okay, so these are actually several different species at about 4 weeks, and here's our human here hanging out at the end. And we can see at 4 weeks, a human does not look crazily different from a cat, a chicken, or a fish, right? They all have heads, right? They've got these tails, okay? We've all got an eye, somites developing, they've got these structures. And then as we move to the next stage, so we'll call this 4 weeks. I'm guessing this is about 6, and we'll call this last one about 8-ish weeks of development. And we'll see again, all right, now the fish is starting to look a little bit different. We probably wouldn't confuse that with a human, but you would confuse a chick embryo with a human embryo, as would with a rabbit or a calf, a pig. You really would confuse these things at early stages. Now at 8 weeks, we start to look a little bit more different. The mammals are really starting to, the human anyway, starting to lose their tail. It's starting to take shape in some of these other animals. And we start to see changes in limb development, other structures, and it is a little bit easier to recognize when we move into 8 weeks. But I just want to point out how similar we are to other species and in our developmental program and patterns, and how we really aren't that different. And I'll tell you what, too, a lot of what was described in the previous week with fertilization and early embryogenesis, those stages of gastrulation and neurulation, where you have those little layers of tissue kind of like folding over and inverting and kind of creating these extra membranes. A lot of those studies are actually done in chicken embryos. Okay, so it's a way that we don't have to research human embryos, and we can, preserve that life and not do those types of possibly unethical experiments, experimenting on, humans. And we can look at those chicken embryos and we can trace where those cells are moving. And that's how we're able to get a lot of that really detailed information on, when does that primitive streak form? Well, how do we know it's mesoderm that's folding in and coming back out on this side? Because of these developmental patterns are so similar. Okay, I just want to give a little quick review of embryonic development, and then I'm going to give you a quick overview of placenta development so that you can then watch the next videos. Okay, so quick review of embryonic development. Again, remember we start with that fertilization moment, the moment that egg and sperm unite. That's going to create a chain reaction and really start our first cell divisions, right? And early cell division is referred to as cleavage because we're really just splitting, we're not growing. And that gets us to the 32 cell staged morula, which is a solid ball of cells around day 4. And then at about day 6, that solid ball hollows out into a hollow ball of cells, the blastula. And this is where things start to get interesting. Okay, and you kind of have to keep both timelines in the back of your mind. What is the potential new embryo doing in terms of becoming a solid ball of cells, a hollow ball of cells, and then kind of flattening out into that little dinner plate disc, which is going to become the embryo. And then you also have to think about what's going on in mom's uterus. And what about the extraembryonic membranes? What are those? Okay, so implantation is happening at around day 7. Okay, and then we're going to get these extra membranes starting to form around day 10. And these guys are going to take a couple weeks to form, as is gastrulation, which happens around day 14, or we'll call it 15. Okay, and that's going to happen as well. Okay, so those extraembryonic membranes, they're starting to form as that hollow ball of cells kind of moves around, and we're creating these different layers. So the inner parts of those layers, that inner cell mass, is not just giving rise to the embryo that will undergo gastrulation, forming those 3 layers, ectoderm, mesoderm, and endoderm, that are going to fold around and become the embryo. It's also making these other layers, okay, that are going to hollow out and create supporting tissues for the baby. So you're going to get a yolk sac. Again, just one more nod back to the fact that we develop very similar to other species like a chicken. Chicken eggs have yolks, and yolk is very important for the development of that chicken. However, for us, we still make a yolk sac, and then it just kind of degrades because it's way less important for a human than it is for a chick embryo. We're going to make our amnion, which is kind of like this nice little bubble that the baby is encased in. And that's going to create amniotic fluid. And that's really what the baby is floating in. So we can see amniotic fluid, right? And just this flat little worm, that's the embryo gastrulating right there. Okay, we'll make the allantois. That's going to be part of the umbilical cord, which is going to be that nice anchoring point that provides a blood supply to and from the placenta so that the baby can get nourishment. And once we start to grow that umbilical cord, that baby can kind of float out and take up more space within the uterus once it's tethered and that structure is created. And then finally, the chorion is going to be formed as well. And this is kind of forming the baby's contribution to the placenta. So we can see the chorion here is really coming from that syncytial trophoblast, this thing that was forming earlier, day 8, that was invading into the mother's uterus. That chorion is going to come from that and form baby's half of the placenta, whereas the mom's half of the placenta is coming from the uterus. So all of this is happening at once, which is why I wanted to kind of break it down and give you the timeline a slightly different way. Okay, so let's get a slightly different view and let's take a little peek at this. I have said most of this already. Again, you will see a yolk sac in early development, which is a nod back to, again, kind of going through those same stages that a chick embryo would go. And then at some point, we're going to go on different paths here. Okay, the amnion, again, it becomes very important for generating amniotic fluid, which bathes the baby and acts as a shock absorber. And eventually it's going to fuse with the chorion. But we really need that umbilical cord to grow and stretch out before that chorion can kind of fill this whole empty space here and fuse with that chorion there. So you can think of it in this picture, we've got, we're at about week 3, and even at 8 weeks, it's not much to talk about. 5 to 10 milliliters of fluid, this is like spoonfuls, as opposed to a liter, liter and a half, right? Like a big bottle of soda. That's how they're usually sold, 1 or 2 liters, right? That's really going to expand and create that fluid. And incidentally, the amniotic membrane and that amniotic fluid, that's the little bubble that breaks when you say, oh, my water's broken, I'm going into labor. And that amniotic fluid is the water that's breaking. So liter, liter and a half is going to come out of the mother's body as a signal that, boop, the baby's breaking its membranes, it's time to go. Okay. And again, you'll watch a much more detailed video on how the syncytial trophoblast starts to develop the chorion, really digs in and invades the mother's uterus so that the baby can get its blood supply. I want to talk about chorionic villi sampling, or CVS. Okay. This is an early test that can be done to get a little sampling of baby cells to tell if baby is healthy and developing normally. Okay. This is a more invasive test than just a regular pregnancy test, or the next thing I'm going to talk about is amniocentesis. So this is the most invasive test that can be done. And you're actually grabbing a little bit of baby cells. So again, we've got baby here. This is about 8-week-old embryo, very tiny at this stage. And again, remember, the chorion is baby's contribution to the placenta. So the chorion cells are baby cells, not mom cells. So you can take a catheter inserted vaginally and just grab a couple of these little cells, and then you can test them. Now, why would you want to do that? Perhaps you're an older mother and you're worried about Down syndrome or other trisomies that could affect development of your baby. Perhaps you have something in your family that is congenital birth defects that may be fatal. Babies born with Tay-Sachs disease. This is common in people of Jewish heritage. These babies have a chromosomal abnormality, and their brain is unable to metabolize fat, and a lot of these babies die a very painful death between 1 and 5 years of age. So if you know you have something like that in your family that might lead to a baby that's going to have developmental problems, baby that might die, have a less than life expectancy, that might be a reason to obtain this information so you can make an informed choice. Why would you not want to get this? Well, you're taking a little chunk of the chorion away from the baby. That's baby's blood supply. If damage is done, if there's bleeding, if the baby is knocked loose from its side of the placenta, you could have a miscarriage. So it's a balancing act between needing information now to make an informed decision about whether you wanted to continue the pregnancy or not, or really risking the whole pregnancy because the baby can be lost with this. Okay, if we wait for the placenta to grow a little bit more, wait for the baby to grow a little bit more, we can see now a lot more development here. That chorion, as promised, has expanded. The amnion, as promised, has expanded out and is almost touching the chorion in a layer. This yolk sac, again, we really don't need it. It's going away. The allantois has developed into the umbilical cord. We can see the artery and the vein coming in here and anchoring into one side of the placenta. Okay. Now that amnion has really filled up like a nice big bubble. And I think this image is so cool. This is a fiber optic view at about 8 weeks. And you can really see that human embryo in there anchored to that placenta and those chorionic villi. It just looks really cool. So now you can see that a little bit clear. And at about 16 weeks, it would be safe to do something called amniocentesis. So this is where we take a little bit of the amniotic fluid. So looking here, so instead of going vaginally, we're able to go transabdominally right through mom's pregnant abdomen, and just a syringe is able to suck out a little bit of amniotic fluid. Again, that's the bubble that the baby's in and will stay in until the water breaks. So there's a couple little like hair or skin cells that might be falling off of the baby. That happens, you don't need that. And if we get enough discarded fetal cells in the amniotic fluid, what you can do is, again, another genetic analysis, just as you would with the chorionic villi. But again, you have to wait till 16 weeks of pregnancy until there's enough fluid there to safely take off a mil or two, okay, so that you can do the test. Again, anytime we stick a needle, there's a little bit of a risk of knocking that baby loose or creating some bleeding or some type of complication. So again, this test doesn't come without risk. People might not want to do it because of the risk associated with the pregnancy. But again, if you have some genetic disorder in your family that might cause a shortened lifespan, again, like Down syndrome, Huntington syndrome, Huntington disease, if you have that in your family and you're not, you don't have the means or capacity to raise that baby, that might be a choice you need to make. Okay, one last thing I want to point out about the placenta and just structurally, because you might come across these words. The placenta is organized into what's described as decidua. Okay, let's use that word. Let's go back to maybe a general biology class where perhaps you talked about deciduous trees. Deciduous trees are trees that shed their leaves during the fall season or whatever season, depending on where you live, right? So deciduous trees lose their leaves. Thinking about the placenta grows and then is shed or lost at the end of the pregnancy. Okay, so thinking about this as a very sheddable organ and a non-permanent organ, we can describe it as a decidua. And I just want to point out the side that is anchoring the baby, this is where the baby decided to implant, again, arbitrarily right here. The baby could have just as easily implanted here or here. This is the baby could implant here. You really don't want to cover over your eggs at the cervix. That would lead to a complication that we can discuss later. But arbitrarily, the baby implants in a spot, and then that's where that anchoring point is. That's where the umbilical cord is going to grow, and that is described as the decidua basalis. Okay, this is where we're anchored to that chorion. Now, there's also decidua capsularis. So think of this as like a capsule. This is more of the free wall. So this is going to be this side here, where that amnion has expanded to come very close to and kind of lay on that chorionic surface, but there's no contact there. Okay, so the side that it's anchored and the side that it's free. And just one last thing to say about the placenta, it can really grow up to the size of a dinner plate. So you're delivering a relatively large organ along with your baby. The baby weighs, 6 to 8 pounds when it's, or I'm sorry, 6 to 10 pounds when it's born. That placenta can be 6 or 8 pounds as well of, tissue weight that you're losing at delivery as well. Okay, I want to come back to this timeline of gestation that was presented in that developmental video for you a couple weeks ago. And feel free to go back to last week's lesson if you want to watch the quick 5-minute video where they lay out all of these nice little landmarks in pregnancy. What I want to do for you here is I want to put on a couple more landmarks, and I want to clarify one thing really quickly too. I have been talking about things in terms of days or weeks, and if you go to your textbook, right, your Martini and Nath, Fundamentals of A&P, this is what you should be reading, and this is where I'm pulling all of my images from. They are very clear to say developmental weeks, because we as scientists speak very different than clinicians. Okay? And that has to do with, we're talking about fertilization, and a lot of what I described to you here as day 1 is day 1 after fertilization. If you go to the doctor, or if you're going to some sort of clinical nursing medical school after you get your bachelor's degree, hey, they're going to talk about last menstrual period as their landmark. So everything that they say in terms of development and where you are in your pregnancy is 2 weeks off. They're counting the 2 weeks from your last period, and you certainly didn't conceive during your period. You needed 2 weeks for that ovulation to happen, right? And this is where we get this discrepancy in timelines. So just be a little bit careful whenever you're reading. Myself and the book are really going to stick to those developmental days. And whenever you're looking at some of these timelines, as we're pulling from clinical sources as well, we're going to be off by about 2 weeks. Okay? So other landmarks that I want to put on here for you. So fertilization, we know that at 7 days after that, that's when we're going to get implantation. Okay, and those membranes, those extraembryonic membranes, don't forget, right? The yolk sac, the allantois, the chorion, okay? Those extraembryonic membranes, the amnion, those we're going to get, it's going to take another week or so after that, right, to grow those membranes, right? So I'm just going to say membranes. And then I think what's on here in purple is very fair as well, because we know that gastrulation is going to happen at about the same time that we're developing these membranes. So I think it's very fair and very accurate, as this figure already indicates that embryogenesis is going to start. And we can also say some of organogenesis. This is when we're going from just becoming layers of cells that are kind of folding and creating that flat little embryo pancake disc into actually creating organs. So whenever you do neurulation, you're actually starting to make the spinal cord and brain part of our central nervous system, right? So that's fair to call that organogenesis. And we'll talk a little bit more about development of heart and lungs and things like that. Okay, so that all comes together. And then what we also need to talk about is what's going on with some of those tests that I pointed out to you. So again, we can do at 8 weeks, but really, it'll be kind of 10-ish on this figure, right? When we cross over from embryo development into fetal development, that's when we can do that chorionic villi sampling. We have enough of the extra embryonic membranes developed, so we could do that. At about week 8 of development. And again, we're not really growing a big enough placenta and amniotic sac until about 16 weeks. Again, I got to cheat on this one and put it at about 18 just because of the way he counts. Okay, so this is when you can do your amnio test. Okay, so again, now that we have these other landmarks on here, we can see chorionic villi sampling is more of a first trimester test, and amniocentesis is more of a second trimester test. Okay. Now, something else I want to point out here too as well, you know, that placenta, it's really forming, and that placenta, you know, it being a full structure, that's happening kind of right at that cross between that first and second trimester. Okay, so that's happening there at about, you know, week 12-ish, right, moving into there. And what's really important about that time point too is this is a really scary time for kind of seeing if the placenta is going to work, seeing if it can fly on its own. Because up until this point, we have the corpus luteum. Remember those little collection of cells that's left over in the ovary after the egg ovulates. That follicle turns into a corpus luteum, and it hangs around for the first trimester of pregnancy, right about till the 3rd month. And it's going to be that primary source pumping out progesterone and something called human chorionic gonadotropin hormone. And that's really what's going to help maintain the pregnancy and not send the ovary and the uterus back into that reset phase where you go through a menstrual period and reset and ovulate. That corpus luteum's got to stick around, and it's got to keep a nice high hormone level. Now, at the 3rd month, crossing over from first trimester to 2nd trimester, that's right about when we lose the corpus luteum, and we hope that the placenta, which has grown out of those extraembryonic membranes from the 1st month, we hope that now that it's had a couple months to grow, that it's going to be big enough and strong enough and enough of a source of hormones for progesterone and human chorionic gonadotropin hormone to carry the pregnancy. So this is often why miscarriage is common in that window. Okay, you're losing the corpus luteum and the placenta just isn't making the right hormones, or there's something wrong there. Okay, and this is often why people don't reveal to their friends and neighbors that they are pregnant till about the 16th week or the 4th month, right? Right when we're doing, we might be doing that amniocentesis to test for genetic abnormalities. Okay, from then on, pregnancy, those signs are going to be pretty obvious. The uterus is going to be pushing out in the abdomen. You're going to be having regular checkups, hopefully with a physician, to make sure that everything's developing on track. Okay, so those are just some landmarks that I wanted to put back on there or add. Okay, now let's talk a little bit more about that hormones that are produced from the corpus luteum and the placenta. Okay, so what's going on here? Now we have it spelled out in case you couldn't quite hear me saying human chorionic gonadotropin hormone. Okay, we will start to produce this at about implantation. That syncytial trophoblast will start to produce some of that human chorionic gonadotropin hormone, and what that is going to do is really communicate to the brain and the mother's body that a pregnancy is in place. Okay, and this is essentially, if you are doing a home pregnancy test, that's what this is here. You put a urine sample on the end of this pregnancy test, or I don't know why we had to use this mysterious discrete blue liquid, but apparently we did. That should be pee, so that's the stick that you pee on if you're doing a home pregnancy test. And what happens here is that hormone, if it's present in your urine, it's going to float down and react with some of these antibodies that are present in the test strip. So if you do have that hormone, that's going to bind to these antibodies, and then that's going to activate some type of coloring enzyme. So you will get a line there. Usually, you got to read the directions. They're all a little bit different, but that's usually what changes the causes the color change on the stick, the presence of that human chorionic gonadotropin hormone. And then there's usually another little line there just to confirm that the test is actually working, that those antibodies are binding there. Okay, so that's your pregnancy test, and it's going to respond to that human chorionic gonadotropin hormone, which you will produce at about the time of implantation, when that syncytial trophoblast starts in very early stages trying to act like a placenta. Again, that corpus luteum is going to be pumping out progesterone and estrogens until this little tiny thing can grow into the size of a dinner plate, right? Our fully formed placenta. Okay, is it possible to get a false positive pregnancy test? Absolutely. One of the craziest examples is the hydidiform moles. What is that? And sometimes this is called a molar pregnancy. You can get implantation without an embryo. So somehow you've got an egg and it finds its way in and it implants, but there was some sort of failure here. Maybe there were 2 sperm that reached the egg, polyspermia was not prevented, or maybe the egg pronucleus died. So this person ends up with only 23 chromosomes, or maybe they have extra chromosomes, 69 or 92. Whatever happened, remember, all of those cell division stages are dependent upon proper chromosome number and proper division. So usually these are all paternal chromosomes, and the egg, somehow its chromosomes died or got lost, and you will get growing of the extraembryonic membranes and the placenta, but this placenta is empty. There's no baby in here. So that's a molar pregnancy. And because you do get the placental structures, you will get the hormones, you will get the positive test, but until you do some type of x-ray or ultrasound, you are not going to realize that there's no baby in the chamber. Strange, but it does happen. Okay, and then one other thing I want to point out with what the endocrine placenta is doing, in addition to making that human chorionic gonadotropin, in addition to taking over the role of producing estrogens and progesterone when the corpus luteum dies at the 3rd month, it's going to take over and start to produce human placental lactogen. So lactogen, think lactation. This is going to get the mammary glands growing. So you will see breast tissue starting to grow and develop, and those mammary glands will start to grow larger and larger in anticipation of producing milk for that baby. Okay, you also have placental prolactin. Okay, that's going to activate human prolactin. This is a hormone that's produced in the brain, right? And that's going to stimulate the anterior pituitary, right, our master gland, to activate and really start making milk from those growing milk ducts. And one thing to point out, at 6 months, those mammary glands are fully developed. Let's go back to our timeline. 6 months, what's special about that and our mammary glands? Well, remember, if we're talking about survival of this baby, right, we had this 50% survival rate coming right between that 5th and 6th months. Whenever you're at 25, 26, 27 weeks, those babies flip a coin 50-50. It's whether they're going to live or die if they were born that early. Again, we want a fully cooked, full-term baby, but if that's not possible, 50% survival, even odds there. So we want mom's mammary glands to be ready in case of any of those early labors and early delivery, because after that 50% survival just keeps going up and up. So the mammary glands better be ready for that baby in the event that it does come early, because odds are it's going to survive. Okay, one final topic for this lesson, and then we'll be done for today. Twinning. Okay, how do we get two babies at once? We are really concerned about developing one baby. What are the differences that need to occur? How often is it? Twins happen in 1 in about 89 pregnancies. The more common type of twinning is the fraternal twin or the dizygotic twin. Dizygotic, 2 zygotes, 2 eggs, 2 sperm, 2 babies that are no more related than any other brother and sister. Okay, and this is the only way that you are going to get babies of different sexes. You certainly could get 2 girls or 2 boys, but this is the only way to get a girl and a boy, because separate sperm, separate egg, separate genetics here. Much more common type of twinning, right? 70%. So in order for this to happen, mom needs to drop 2 eggs whenever she ovulates, and then 2 sperm. Obviously, there's plenty of sperm to go around that will fertilize. Some of this is inherited, so sometimes people have twins that run in families. And sometimes this happens to older mothers. Remember, mom's going to run out of her egg supply at age 50. So as we're nearing the end, that ovary's like, let's get rid of these eggs. It starts dumping extra. So sometimes older mothers end up with twins. What a nice surprise right before you go infertile. Okay, the identical twins, monozygotic, 1 zygote. This needs to happen very early in pregnancy. So you start with 1 zygote, okay, and then somewhere in that cleavage, morula, blastula, and then maybe a tad bit into implantation. Really need to get this done before day 12 before we start to build our membranes. Okay, then this is when you can get identical twins. After day 12 happens, nothing's split, and this can't happen anymore. Now, what if you get an incomplete split? This is rare, and then this is where we might get conjoined twins. Okay, so our famous twins that hail from Siam, right? These guys really made that famous. A lot of times conjoined twins are not viable because of some of the ways that the babies split. It's really challenging for these individuals to move around and live their lives. But the first really famous set of twins from Siam, these 2 individuals lived a very long and healthy life. Okay, we've already talked about chimerism and gynandromorph. This might be where you have kind of a ball of cells in your body that maybe was of that identical twin, and then maybe those cells got reabsorbed, or something went on during development. Again, those we typically don't see very commonly, or if you are a chimera, you probably don't know, unless there's some strange crime scene thing where you're, DNA testing a bunch of cells and tissues at the crime scene. Anyway, let's get back on track here. So looking at the top part of this panel, panel A, this is going to be the dizygotic fraternal twins. Again, 2 eggs, 2 sperm, 2 zygotes, and they implant in 2 separate spots in the uterus, and they're going to get 2 amnions, 2 chorions. They get their own room, they get their own beds, they get their own space. Okay, so those are going to be very separate. Now, if you are an identical twin, you're going to have to share a lot more. So 1 egg, 1 sperm, as we start to go through cleavage, something happens here. So now we have two inner cell masses. So that means two babies, right? Two embryos. Okay, so the single structure will implant. Okay, and you will make only one placenta and one chorion. You will make two amnions. So you get your own bed, but you don't get your own room, right? We're sharing here. Okay, and that's really what goes on with twins. Okay, that's enough for today.